Well, good afternoon to you, and welcome to the web viewers at the various UC campuses. My name is Rick Keel. I'm Chair of Electrical and Computer Engineering here at UC Davis. And I'm very happy to be able to introduce our speaker today. I'm delighted that Professor Simon Cherry is here. Professor Cherry received his Bachelor of Science in Physics and Astronomy from the University College of London in 1986. He received a PhD in Medical Physics from the Institute of Cancer Research at the University of London in 1989. Then in 1993, after a postdoctoral fellowship with Edward Hoffman at UCLA, he joined the UCLA faculty in the Department of Molecular and Medical Pharmacology. In 1998 to 2001, he was Associate Director of the center there, the Crump Institute for Molecular Imaging. And then in 2001, he joined us here at UC Davis as Professor of the Biomedical Engineering Department and Director of the Center for Molecular and Genomic Imaging. Dr. Cherry was also served as Chair for BME here at Davis from 2007 to 2009. He has a number of distinctions. I'll just mention a few. He was a founding member of the Society of Molecular Imaging. He's a fellow of the IEEE and the AIMBE. He gave the Henry Wagner Distinguished Lectureship at the annual meeting of the Society of Nuclear Medicine in 2006. And he received the Academy of Molecular Imaging Distinguished Basic Scientist Award in 2007. He's also co-author of the third edition of the textbook, Physics in Nuclear Medicine. Will you please help me welcome Professor Cherry?
Well, thank you, Rick, very much for the kind introduction. It's a pleasure to be here. I'd like to welcome everybody and also all the people viewing at our sister campuses today. And the topic I'd like to address today is some of the advances in medical imaging, and in particular the idea of integrating different imaging modalities together. And the two modalities we're going to talk about today are magnetic resonance imaging, or MRI, and positron emission tomography, or PET scanning. So to motivate this a little bit, if we consider the range of imaging modalities that we have accessible to us today, we have techniques such as X-ray computed tomography and magnetic resonance imaging and ultrasound that are very good at portraying structure and morphology. They give us great insight into anatomy. But a lot of disease processes start out not with anatomic change, but with some kind of biochemical or molecular abnormality, or even changes of the expression of the level of gene expression. And so, as we move forward into this era of personalized medicine, it's also going to be very important to have techniques that give us more functional information, that can look at physiology, can look at metabolism, and can look at specific molecular targets, be that a receptor on a cell surface or an enzyme that catalyzes a specific biochemical reaction inside of a cell. And in that arena, techniques that use probes that are contrast agents that are exogenously administered, such as for optical imaging, you could use fluorescently labeled agents, or the techniques we're going to talk about today where we use radioactively tagged compounds to elucidate some of the molecular mechanisms and molecular targets inside of the living body. In doing that, we get great insight into specific biological processes or parameters, but we also still need to know exactly where those are taking place. And so this idea of combining structure and function is very important. Having techniques that give us high-quality structural information, such as X-ray computed tomography and magnetic resonance imaging, combined with techniques that tell us something about the function going on inside the body as well. And so that's the topic I'm really going to address today. And the two technologies that we're going to try and merge are magnetic resonance imaging, which is very strong at anatomic imaging, with PET scanning that's very good at molecular imaging. So before I talk about how we combine these techniques, let me give you a little bit of background about both of these technologies. And this will be just a very brief overview. So let's start out with MRI first of all. So MRI, we're essentially imaging the signal coming from protons inside of our body. And in the absence of a magnetic field, these protons, they have a property known as spin, and the orientation of this spin vector is random. But when you put a sample inside a strong magnetic field, the spins on the protons either line up with the field or against the field. And so you have these two different energy levels that are actually quite close in terms of energy, and we'll talk about that in just a moment. And you have a slight excess of spins that are parallel to the field versus those that are anti-parallel. The energy difference here is given by a constant times the magnetic field strength, and if you do the calculations, you'll find out this ends up in the radio frequency part of the spectrum. So it typically in the range of tens to hundreds of megahertz is the frequency corresponds to that energy difference. So if we want to take spins in this state and move them up to this energy level, we have to put radio frequency radiation into the sample to make that happen. And that's what we're going to do in MRI. We're going to give radio frequency pulses to move spins into that more excited level. And then the rate at which those spins come back down to the parallel state again tells us something about the environment in which those spins reside. And that's the basis for the signal that we look at in MRI. How do we get position information? Well, you notice this energy level depends on the field strength. So if we can arrange for the field strength to vary slightly across the sample, that means different radio frequencies will excite spins at different locations in the sample. And therefore we can encode some spatial information. Now we have whole graduate courses on MRI physics that would take up many hours to describe the details of how MRI works. But that's the basics beneath the signal generation. And then if we look at the components of an actual MRI scanner, we have three key components that we're going to be interested in. First of all, we have a very strong magnet. That creates the main magnetic field that creates these two energy levels with the spins either parallel or anti-parallel to the field. We then have a set of gradient coils that impose a gradient on that magnetic field. This is what's going to give us our spatial encoding. It makes the magnetic field change slightly across the sample so that the radio frequency we need to excite spins depends a little bit on the location of where those spins are, and that's how we're going to get an image. And then we have radio frequency coils that pulse in the radio frequency radiation to excite the spin sample. And then they also receive the signal back as the spins de-excite. So in the context of today's talk, the thing to think about is that we have three key components inside an MRI scanner. We have a strong magnet. We have gradient coils that give a smaller magnetic field, but give a gradient in that magnetic field, and then we have radio frequency coils. So MRI produces exquisite images of the anatomy, and here's some examples. We routinely get millimeter level spatial resolution in clinical studies. If we go over here, this is a cross-section through the human brain. One thing that MRI does very well is to give contrast between white and gray matter, because the relaxation properties of the spins in those two tissue types are slightly different, so we can exploit that to get contrast. Compare that, for example, with X-ray techniques, which look at electron density, where it's very hard to see differences between gray and white matter. And this actually is an image of a patient with multiple sclerosis. It's a disease of the white matter, and so these areas of enhanced contrast here relate to the pathology. Here's a cancer example. This is a breast imaging example with a primary breast tumor. In this case, some contrast has been given to the patient. Contrast agents for MRI typically involve the use of the element gadolinium, and that changes the relaxation parameters of these spins very dramatically and leads to changes in the signal intensity that can result in contrast. So very beautiful structural definition of what's going on inside the body. Doesn't tell you much about function, though. So let's switch to the second technique that we're going to talk about, which is positron emission tomography. And I have a short movie here to illustrate this. And in this case, what we're going to do is we're going to take a compound of interest and we're going to tag it with a radioactive tag. So here are all the radioactively labeled molecules sitting in a syringe that then get injected intravenously into the patient. These radioactively tagged molecules travel through the body trying to find their target. These are usually targeted molecules. They're trying to find a specific receptor or interact with a specific enzyme. Here I have just one radioactive molecule about to decay. It decays by emitting a positron. That positron travels briefly through tissue before it meets up with an electron, and the electron and positron annihilate, producing two back-to-back 511 kiloelectron volt photons. And so the PET scanner is trying to detect these two high-energy photons in coincidence emitted back-to-back as a signature that a decay event happened inside the body. And so a PET scanner is typically a ring of detectors that surround the patient, such that if we have a radioactive decay occurring inside the patient, we get our two annihilation photons produced, and we're looking for detections on opposite sides of the body at the same time. That's one event. A PET scan consists of acquiring something on the order of tens to hundreds of millions of these decay events to actually form ultimately an image. And in fact, we don't directly get an image. We have to use mathematical reconstruction techniques, the algorithms of computed tomography, to actually go from the raw data to a reconstructed image. Now, the strength of PET imaging is in the chemistry, in what we can inject into the patient and what that can tell us about the function. So first of all, let's just think a bit about the isotopes we have available for tagging things with. And for PET, we need something that decays by positron emission. That's why it's called positron emission tomography. And so I list just a few of the isotopes that are available to us here. The most widely one used clinically is this one, fluorine-18. Has roughly a 2-hour half-life. Obviously, for anything biological, having an isotope of carbon is very nice. So carbon-11 is a positron-emitting isotope. So just about any naturally occurring, interesting biomolecule you can think of is going to be organic and is going to have a carbon atom in it. With PET, what you could do is you could take that stable molecule, take out the stable carbon-12, put in a radioactive carbon-11 atom. You've not changed its biochemical properties in any way, but now it's labeled so you can actually image it. The problem is, as you can see, the half-life of carbon-11 is only 20 minutes. That means you have to produce it locally in an accelerator, a biomedical cyclotron, on site. You then have to do the chemistry to put this carbon-11 into whatever compound you want it to be in. You then have to get it injected into the patient and get the imaging done, all in a few half-lives. So typically, 2 to 3 hours for carbon-11 from the point you produce it to the point you finish imaging. So it's very time-intensive, very labor-intensive, and very challenging in a clinical environment to do that, which is why fluorine-18 is much more widely used clinically. 2-hour half-life. You don't have to have your own accelerator. There are companies that distribute this material regionally, and so this has been the mainstay, and I'll show you the compound that's actually used clinically. We do have some longer-lived things like copper-64, 12 hours, iodine-124, 4 days. And for some studies, these are of interest, but they're not so biologically compatible, obviously. And actually, having a short half-life from the patient's perspective is a good thing because it means the radioactivity decays from their body very quickly. They don't remain radioactive for long after the study. So that's a good thing. So we have these isotopes. Now, what can we do with them? Well, we can label all kinds of different things. And if you look in the literature, it's just an amazing variety from simple ions, such as just using fluoride iron, which is avidly incorporated into bone and gives you beautiful images of bone metabolism, through small molecules like labeling drugs, pharmaceuticals, understand their biodistribution, substrates for enzymes, so you can measure enzyme activity, ligands for specific receptors, so you can look at receptor distributions in the living body. You can then go to larger biomolecules like peptides and antibodies. There's a lot of interest in these from a therapeutics perspective. There's areas like looking at gene therapies, and there are now approaches where you can actually image therapeutic genes using radio-labeled probes. You can tag cells, so this whole growing area of stem cell therapeutics. You can label cells with a radioactive tag, so you can follow those in the living human being. We can look at distributions of pathogens, such as viruses and bacteria. And there's a lot of interest in nanoparticle therapeutics and diagnostics now, and of course we can label those and look at those too. So really your imagination is the limit in terms of what you can label, what you can then look at inside a living animal and eventually a living human being. So the most common use of PET clinically, if we switch to just the clinical use rather than research use, is this compound fluorodeoxyglucose that's shown on the left here. So this is an analog of glucose. It is labeled with fluorine 18, which will allow us to visualize it in a PET scanner. And it gives us images essentially of glucose metabolism. And so here I show what a PET scanner looks like, and here I show some images from a cancer patient obtained after injecting this compound into the body. So this compound goes everywhere, and it's giving us an image where the intensity is roughly proportional to the use of glucose. Glucose, as you probably know, is one of the major ways that we produce ATP in the body, which is the energy currency of the cell. So it's very, very important. So the brain is outside the field of view here, but the brain is one of the most metabolically active, most energy-demanding organs we have, and so the brain takes up a lot of this radioactive glucose. The heart, likewise. You see here the heart quite clearly. You also see some uptake in the liver. And this compound is excreted through the kidneys, which you see here and here, into the bladder. But the worrying thing in this particular patient's case are these hot spots that you see, these little round spots of increased activity where there's no organs and you really shouldn't be seeing anything. And this is all metastatic cancer. So cancer cells rapidly proliferating, have high energy demand, therefore they also use a lot of this radioactive glucose. And so this has become a very effective way for screening the whole body for staging of cancer patients. So perhaps you've already identified a patient has a primary tumor. The big question for treating a patient is, has it spread? And PET scanning is now routinely used to answer that question. So that's a little bit about PET in the clinic and MR in the clinic for structural imaging. But a lot of our focus in my lab and in many labs across the country has not just been in human imaging, but has also been in research applications in animal models of disease. Because what we're really trying to do is we're trying to develop the next generation of diagnostic techniques, and of course we have to start in animal models to do that. And so imaging is a very powerful technology, not just in the hospital setting, but also in the biomedical research setting. And these days we have just tremendous opportunities to develop new approaches to either diagnose or treat disease based on our understanding of the sequence of the human genome, based on combinatorial chemistry techniques that allow us to screen millions of compounds against a specific biological target to find those compounds that will interact with that target. And this throws up many opportunities for new therapies and diagnostics, and the question is, which ones of these are you going to take into clinical trials in humans? Because doing clinical trials in humans is very expensive, very time-consuming, and failures, unfortunately, are all too common. Often for toxicity reasons, things fail well into clinical trials after you've spent years and billions of dollars trying to develop these compounds. So how do we better streamline and better predict which things to take into clinical trials that are likely to work well? And so one argument is you go in vivo into living systems as soon as you can. You don't just rely on cell culture. You move into animal systems early, and you use imaging as one of the techniques to monitor your new drug, because we could radio label it and we could see exactly where it goes in the animal. Not just does it go to its intended target, but where else does it go where it might cause toxicity and side effects. And so now all the drug companies have their own imaging centers. They have PET, they have MRI, they have all these technologies now in-house for small animal imaging for this reason. And so just like we have scanners for human imaging, we have scanners for small animal imaging. We can get resolution of 100 microns or so with MRI in mice, and here's just a few examples. This is a particularly nice image here of the mouse brain where you see the structure of the cerebellum really beautifully. This is all non-invasive imaging. Looks like the mouse was cut open on the left here, but this is just all software done. We acquire a volume of image data, which we can then interrogate using advanced image processing software however we would like. And then we also have PET scanners for small animal imaging as well. This is a mouse with cancer. It's carrying a model of human breast cancer. And this is one of the mammary fat pads of the mouse down here. And here we see this is the same compound, that fluorodeoxyglucose compound that we were using in human cancer imaging, now used in a mouse. And so just to orient you, this is the heart, these are the kidneys, here's the bladder, but here's the tumor we're interested in. This mouse was treated with a chemotherapy. You see the tumor goes away 1 week later. Another week later, it's still gone. The mouse was then taken off the drug. We continued to monitor the mouse, and you see that another 2 weeks later, the tumor comes back. often happens clinically as well. The mouse then went back on the drug a second time. The tumor continued to grow, suggesting that the clone of cells that had grown in this animal were now resistant to that particular drug. And so this is just an example of being able to monitor through imaging the initial development of a tumor, its regression with treatment, its recurrence, and then its resistance to that treatment. So you've seen individually how we can use PET and MRI to look at various things. And so now the question is, if we could merge these together into a single instrument, do we end up with something that's a really powerful research tool with which we can do some unique science? Or do we just end up with a very complex, expensive instrument that really doesn't buy us anything new? And so that's the question that we've, in our lab, have been trying to answer for the past few years. So first of all, why do this? Well, because we would like to have both structural information and molecularly specific information from the same subject at the same time with perfect registration between them, so we can really understand what we're looking at and what we're trying to measure. And one of the things I've not had much chance to talk about is that MRI actually is a lot more than just structural imaging. It gives you beautiful anatomic imaging, but there's a lot of physiologic measurements you can do with MRI as well, such as looking at diffusion weighting. You can look at dynamic contrast enhanced MRI using contrast agents. There's MR spectroscopic techniques. There's ways to label cells with MR and track them. There's also a whole host of people developing MR-specific molecular imaging agents as well. So combining those more advanced MRI techniques with anatomic MRI, with PET, all in the same imaging session would be very attractive from a research perspective at least, and maybe also clinically important, and we'll come back to that towards the end. Now, the weaknesses are, as I said, this is going to be difficult to do. These are two very different modalities, as you will see in a moment, and there is this question about what the ultimate throughput would be clinically, cost effectiveness, and what the role would be. So what are the technical challenges of doing this? What we're trying to do here is we're trying to take a PET scanner, we're trying to take an MRI system and put them together so we can get images at the same time from both systems. So we're going to worry about the ways that these systems may interfere with each other. And so things that are obvious are, well, the MRI has this strong magnetic field. So what we use for our detectors for our PET scanner better be fairly immune to those strong magnetic fields. We also have to worry about electromagnetic interference between the MRI and the PET. And it goes both ways. The MRI, where we're pulsing strong radio frequency radiation to excite those protons inside the body. The PET electronics would pick that up and it could completely distort the PET signal, and vice versa. We've got all this electronics reading out our PET detectors that's generating radio frequency that could easily interfere with the MR as well. And then we have other issues just such as space. Magnets are expensive. The bigger they get, the more expensive they get. So magnets tend to be only just slightly bigger than the subject you're trying to image. So now we're trying to get PET inside of that magnet as well. As you'll see, space is a major constraint. So there are two fundamentally different approaches you could take to this integration. And the one on the left here is the easiest, perhaps though the least interesting. And that is, you actually keep the systems physically separate. You have an MRI scanner, and right outside of the MRI scanner you bolt a PET scanner to the floor, and you have a common patient bed that moves through the two scanners, and you have some nice software that integrates the two. So this I'll call the tandem approach. The advantage here is it's much easier to avoid interference because you can shield to a certain extent the electronics and the detectors in the PET scanner from the magnet. The big disadvantage from a research perspective is you cannot take the data at the same time from the two modalities. So you've just lost one of the key reasons for doing this whole thing, which is to get data at the same time. Living subjects are dynamic, things are changing over time, and particularly if you're going to make measurements of functional parameters. These are not static in time, and therefore you really want to take the measurements at the same time. So we and many others prefer this approach, where we're actually going to build the PET scanner inside of the magnet so that both scanners are imaging the same field of view at the same time, and we take data at the same time. And this, of course, is much higher throughput as well because you don't do the things sequentially. It gives you the best registration of the two data sets, but it's also technically the most challenging. So the geometry that we're looking at here, we're now taking a cross-section through the magnet, and we're focused initially in this prototype on preclinical imaging. So everything I'm going to show you in the next few slides relates to trying to do this in mice. And so here you see the magnet on the outside. Inside the magnet, we have the gradient coils that we talked about on the MRI scanner. Now we're going to place our PET detectors inside of that, and then inside of that is the radio frequency coil of the MR system, and inside of that is the subject that we're trying to image. Now, to understand the choices that we've made, we need to talk a little bit about the detector technology that's used in PET imaging. How do we detect these high-energy photons that are given off from the radioactive decays that we are trying to image? And typically, we use scintillation detectors. So these scintillators, these are dense crystalline materials that when a high-energy photon or gamma ray interacts in that material, it produces a flash of visible light. And we then use something called a photomultiplier tube to convert that visible light into electrons and then actually amplify those electrons by about a factor of 1,000,000 to give a nice current pulse coming out the bottom of our detector. So it's kind of a two-stage detector. The scintillator converts the high-energy photon into visible light, and then the visible light is converted into electrons by the photomultiplier tube. Now, the photomultiplier tube is a vacuum tube, and the electrons are traveling some distance through this tube. So guess what? You put this in a magnetic field, and bad things happen. Because the electron trajectories are perturbed by the magnetic field, the electrons don't go where they're supposed to go, and the signal coming out the photomultiplier tube drops very rapidly as you increase the magnetic field strength. So you see that you're okay for a few tens of Gauss magnetic field strength, but as soon as you go much above 100 Gauss, then the signal starts to drop very precipitously. We're talking hundreds of Gauss here. A 7 Tesla MRI system is 70,000 Gauss, so way off the scale here. So photomultiplier tubes will not work at all in that environment. So we need a different detector to convert the light into electrons. And so the detector we use is an avalanche photodiode. This is a thin silicon detector. It's position sensitive, so we're able to sense where the light hits the detector. And the nice thing is, because the multiplication takes place in a very thin region with a very high electric field across it, it is pretty insensitive even to the magnetic fields you find inside of an MRI scanner. And so we're going to replace the photomultiplier tube with these avalanche photodiodes, and therefore have a detector now that can go inside of the magnet. So here's a view of what our detector looks like. We have an array of scintillation crystals still. This is what's going to sense the high-energy photons and convert them into light. We then take that light through a short fiber optic bundle, which is shown in white here, to the avalanche photodiode that decodes that light, tells us where it was, and we have some electronics here to read the signal out. This is all made as much as possible from non-magnetic components. So that's one detector module. As you'll notice from the earlier picture, we need, of course, a ring of these around our subject because the photons are coming off in random directions, 180 degrees apart. So we need to if we want to capture them all, we have to surround the object with detectors. And so here's now the actual imaging device that we developed, which is made up of 16 of the detectors that I just showed on the previous slide. So the ring of scintillator elements sits here. You see the optical fiber bundles coming off to the sides. 8 go one direction, 8 go the other direction. Here's the avalanche photodiodes here, and then the electronics to read them out. We then use some shielding over that electronics to try and minimize interference. And then this whole insert, which is about 50 centimeters long, can then slide inside of the magnet. And now we have our combined system. So here's a 7 Tesla animal MRI system. Our insert is now deep inside the bore of this system. We have some cables coming out to bring the data out. We also have a cable going in to supply the bias voltage for the detectors. Here's the electronics and the computer that control the acquisition of the PET data. The MRI here is just a commercial MR system. We have not modified that in any way. What we've done here is we've built a PET scanner that's compatible with a commercial system and built it into the scanner. Only a few very technically oriented people will be interested in the precise details of the number of crystals and the field of view. Suffice to say, the field of view is large enough to image mice, which is what it was designed for. And this is a fairly state-of-the-art MR system with a 7 Tesla magnetic field and pretty good gradient strength. So now we've got the system. The question is, well, does it work? Do we have interference between the two modalities that is going to degrade the images or not? And so we've run a lot of tests to look at this, and I'll just show you a few of the numbers. And so this test is looking at one detector module, These spots here each correspond to one of the crystals in a detector module inside the PET scanner. And so what we're hoping for is that we can resolve all the spots. That means we can see all the crystals. And indeed we can. And the other thing we're looking at is pulses on the oscilloscopes. This is the raw signal coming out of the PET detector, just to see if we see any baseline shift, any introduction of noise. And so here we're inside the magnet, but we're not actually running the sequences that generate the radio frequency and the gradients. So the MR is effectively silent, although the magnetic field is there. And now we're starting to run kind of standard MR sequences, spin echo, gradient echo sequences. The pulses look fine. These maps, these detector maps also look fine as well. So that's encouraging. That's looking at the raw data. If we now go to looking further down the line, we look at reconstructed spatial resolution of the PET data, we see that it looks fine no matter whether we're running the MRI sequences or not. We have an image resolution in the PET scanner of about 1.2 millimeters. The one place we did see a small effect was when we looked at the absolute number of events detected per unit time in the PET system while we were doing various things on the MRI. It's a small effect, and it's not statistically significant, but there's definitely a trend here that you can see. So these three columns here refer to pulsing the gradient coils in the X and the Y and the Z directions at two different power levels, 100% power, which is the most demanding, and 50% power. And you notice there's a trend that when we're at the higher power levels, we seem to be getting slightly lower counts per unit time on the PET detectors than when we're at the lower power level. And there's a definite trend that way. Notice this scale is greatly expanded, so it's a very, very small effect. But still, it's something we would like to investigate and understand a little better. It has no noticeable effect, though, on the actual images that we get from the system. Now, one nice little physics advantage of doing PET scanning inside of a magnet is a slight improvement in the spatial resolution of the PET component for the reason I'm going to explain here. So if we go back to this still shot that came out of the movie I showed earlier. So here's our radioactive molecule with a radioactive atom here that's in the process of decaying. And what we want to image is where those radioactive molecules are in the body. That's the whole point of doing a PET scan. We want to see where those radioactive molecules are. But remember, what actually happens to actually get the data that forms our image is this atom decays. It kicks out a positron. The positron travels a little ways through the tissue, scattering off the electrons until eventually it slows down enough that it can combine with an electron in the tissue and produce these two back-to-back high-energy photons that we ultimately detect. So what we actually image is not where this molecule is, but it's where these annihilation sites are. So there's a slight error in the image, and that error is called the positron range. It's effectively this distance shown in red here. And as you might imagine, this gets worse the more energetic the positron is that's emitted. So if you get a very energetic positron emitted, it's likely to travel further before it annihilates. Therefore, the positron range will be longer, and the mispositioning of the event will be worse. That's illustrated here. So I've got 4 different isotopes here. I'm showing the highest energy positron that these isotopes can produce. And then I'm showing the point spread function, or the blurring, if you like, caused by this positron range effect. So fluorine-18, which has a fairly low energy positron on average, you can see there's not much blurring. This is a millimeter scale down at the bottom here. But if we go up to oxygen-15, which has much more energetic positrons, now you see the positrons go much further before they annihilate, and so we're going to have more blurring in our image because of this. So why is all of this relevant to doing this inside of a magnet? Well, a positron is a charged particle. Now we're going to put this inside of a magnet, and so the positron now will not travel as far before it annihilates on average as it would have done without a magnetic field. So here are some simulations showing a point source of a radioactive source here at the center. You see the positron tracks. You see the positron tracks straggling off in all directions here when you've got no magnetic field, and that's the positron range effect. But now if we put this sample inside of a 7 Tesla magnetic field, the positrons spiral around the magnetic field lines. They feel that force, and on average they don't travel as far before they annihilate. So it tightens up that point spread function, and we have less mispositioning because of positron range. Now, for fluorine-18, the effect is virtually undetectable because they have very low energy positrons to start with. But if we go to one of these more energetic positron emitters, like bromine-76, what you see a sharpening of the point spread function inside the magnetic field, which is shown in the profile in blue, compared with outside the magnetic field, which is shown in red. So for high-energy positron emitters, we're going to get a slight improvement in spatial resolution with PET inside of an MRI scanner than separately. So that's one nice little side advantage of doing this. So now what about, we've looked at the effect of the MRI on the PET. The PET seems to be working fine, no problem. What about the other way around? Are we affecting the quality of the MR images? So we ran some standard test studies, looking, for example, at uniformly filled cylinders and looking at image uniformity and signal to noise. And those results are summarized on this slide here. Signal to noise on the left, 3 different pulse sequences, the PET inside the magnet and no PET scanner there. So you see we do reduce the signal to noise just very slightly with the PET scanner inside the magnet, and we also reduce the uniformity of those images a little bit, but these are very, very small effects and actually surprisingly small considering all the material and electronics we have placed inside the bore of the magnet. And a final test I'll show you, which is a more sensitive one to look at the noise background, is to look at the subtraction of 2 sequential images acquired under identical conditions and then looking at the noise pattern that's left when you subtract them. And so in this case, this is the control. The PET scanner is off in both cases. So this is kind of just the background noise of 2 sequential MR acquisitions. If we then do the pair of the PET on minus the PET off, we see no increase in the noise background, suggesting that the PET really is having no effect on the MR system. So all of that looks good, and so that means we can now actually start to use this for some actual applications. And this was one of the very first studies that we did, where we simultaneously now, for the first time, took MRI and PET data in a living subject. In this case, it's a mouse that has a tumor growing inside. You see the structural image down here. These are transverse sections through the mouse, just a couple of different levels. This is the tumor here. And here's the PET scan using that same fluorodeoxyglucose compound, so the tumor is metabolically active, as you can see. Interestingly, you'll notice there's a region in the tumor that appears to have very low metabolism, and that also corresponds very nicely when you overlay the images on this bright area here in the MRI scan. So what's that? Well, that bright area is almost certainly tumor necrosis. It's a region of the tumor where the cells have died. That gives high intensity on MRI, and therefore it shouldn't be metabolically active because the cells are dead, and indeed you see a hole in the metabolism signal at that location. And so having both the metabolism and the structural data at the same time allowed us to really understand that signal and to be sure about what we were seeing in what's otherwise a pretty heterogeneous signal, both on the MRI and on the PET. Now, some more advanced things are shown on this slide. So here we've got 3 different studies done in the same session. We have an anatomic MRI that's shown in the grayscale. Again, this is in a mouse that has a tumor in the flank. This is the tumor here. So we see the tumor very clearly. We've then done a glucose metabolism PET scan, and that's shown in the red scale here. So you see that part of the tumor is very metabolically active. There's other parts of the tumor that are not. And we've then done a more advanced MRI sequence called diffusion-weighted MRI, where we're looking at diffusion of water through the tumor. And so if the tumor is densely packed with cells, water will not diffuse very fast, whereas if the cells have died and you don't have a lot of cell membranes to cross, the water will diffuse more rapidly. So interestingly, again, you will notice in the region of the tumor that on average has higher diffusion, meaning you've probably got cell death here, you have lower metabolism. And again, that makes a lot of sense. The last example I'll show from our work is again combining 3 different things. And it's again a mouse tumor model. Again, we have a large tumor in this animal shown on MRI in the grayscale. And as you're getting used to by now, we have the glucose metabolism image by PET, which once again shows that we have some very metabolically active regions of the tumor and some other regions of the tumor that are not at all metabolically active. The standard approach before we had this combined instrument for MRI would have been, okay, we want to know more about this tumor, so we can see the tumor on the MRI scan, so we will define a region around the whole tumor, and then we will do something called MRI spectroscopy, which gives you some information about the chemical composition, some of the major constituents in the tumor. So, for example, we can look at choline and creatine components inside the tumor. Now, if I'd averaged that from the whole tumor, I would have got a very misleading result because the tumor is very heterogeneous in this case. With the PET now, I can actually guide the spectroscopy to do it in different regions of the tumor. For example, a metabolically active part of the tumor, which gives me a spectrum like this. This is just a background muscle region. And now if I go to a region of the tumor that's not metabolically active, you'll see that the peaks for creatine and choline are now quite different. And indeed, areas that have high fluorodeoxyglucose uptake have a high choline to creatine ratio. Areas with low uptake have a low choline to creatine ratio. Choline is known to be associated with membrane turnover rate and cell proliferation. So again, the biology here is all making sense, and we were able to actually guide regional spectroscopy using the metabolic signal. So what is the relevance of all of this to the clinic? Well, we now also have human combined PET MRI scanners as well. So doing the kinds of studies that I've just shown you in animals is now a reality in humans. Two of the major medical imaging corporations have prototype systems out in the field now. This happens to be the system from Siemens that's installed at Mass General Hospital. So this is their 3 Tesla human MRI scanner. This is the PET scanner insert that inserts mechanically inside of the magnet. And these are images of the human brain taken with PET and MRI at the same time. And then Philips are also developing a system. They're taking a different approach. They're taking the tandem approach where they actually keep the 2 scanners separate and do the studies sequentially. So we're now moving this into the clinical diagnostic arena. And so the question is, well, what are we going to use it for? And so clinically, there are many applications, first of all, where MRI is the preferred anatomic imaging modality to X-ray methods. So, for example, the abdominal region is an area where MRI is very good, and certainly in the brain as well. Anywhere where you need really good spatial registration. So you might think, well, why can't you just take the scans separately and use software to register them together? In the brain, that works pretty well, but in the rest of the body, organs move around. The way the patient lies on the couch will be slightly different in the 2 scans. It is very, very hard to accurately register these 2 data sets if they're not taken at the same time in the same geometry. The things we're interested from a research perspective in doing are some of the later things in the list. So correlating functional or physiologic MRI measurements with the anatomy and with PET. Some of the later examples that I showed you were indicators of that. And then there's some interesting things we can do with the chemistry as well, with interesting isotope pairs. So fluorine-18, as you've seen, is a PET radioisotope that we can use for imaging. Fluorine-19 gives pretty good signal on MRI. So you could imagine doing a fluorine-19 spectroscopy and fluorine-18 PET at the same time. We also have pairs for carbon-11. C13 gives MR signal. Carbon-11 is a PET radioisotope. So there's probably some very interesting things that we can do with these radionuclides. So to summarize, prototype combined PET MRI systems have been developed both for small animal and now also for human imaging. There's a whole range of interesting things going on the chemistry side as well that I haven't had a chance to discuss. Building, for example, nanoparticle platforms that are both have a PET contrast agent and an MRI contrast agent on them. We think that this combined modality is a very powerful platform for biomedical research, and as you see in the translation for clinical applications is just starting to be pursued. Now, to develop this system was a huge team effort that's taken many, many years. I'd particularly like to acknowledge my former graduate student, Ciprian Katana, who's now at Mass General, and particularly Dr. Yibao Wu, who's sitting in the room over here, who's done all the testing on the current insert and is actually developing the next generation system. We've had a very good collaboration with Caltech, who've helped us a lot on the magnetic resonance imaging and on the animal studies. And we also have collaborations with industry for the avalanche photodiodes and former co-workers who are here at UC Davis and then have gone off and established their own group now at the University of Tübingen. And so with that, I'd like to thank everybody for their attention. I'll be happy to answer any questions. Thank you.
Questions?
You mentioned magnetic shielding on the electronics of the PET system. How do you accomplish that?
Okay, so that's a high-frequency copper laminate. That is effectively shielding the PET electronics from the hundreds of megahertz signal from the MR. What we've discovered more recently is that there's a second source of interference at much lower frequency that comes from the gradient power supply, and that copper will not shield that. And so we're looking at other ways to shield that.
Other questions? Go ahead.
What kind of, it was difficult, I guess, to find all the non-ferromagnetic materials for this?
Yeah, so you have to be careful what you use, obviously. And one of the other challenges is just what's available, particularly in terms of electronic components. So the biggest issue for us has been, one of the biggest issues has been on the preamplifier side. We use a commercial preamplifier, and so we've been trying to work with that company to develop a completely non-magnetic version of that. But we really haven't seen a lot of interference effects. And we're actually surprised that they're as small as they are, given what we've put inside the magnet. And one of the interesting things will be when we scale up with the next generation system that I didn't talk about that we're currently building, those effects may become a lot more noticeable. So we have an ongoing study that's really looking very carefully at shielding, and also we're thinking obviously very carefully about what materials we use in the next system.
Question in the back?
Yeah.
How does the radiation dosage compare to CT?
Yeah, so that's a good question, and it's not one where you can give a single answer, unfortunately, because it depends so much on the examination. So the radiation dose distribution is quite different. So with CT, if you know that you want to look at the liver, for example, you just take a CT scan over that part of the body, and that's the only part of the body that gets a radiation dose. With PET, it's a systemically administered radiotracer, so it goes everywhere in the body. So you're automatically getting a whole body dose. So the doses from PET typically are on the same order of magnitude as CT or slightly higher. But they're certainly still very much in the diagnostic regime. People have multiple PET scans, multiple CT scans over a period of time, quite routinely diagnostically. You may know that, and I didn't really discuss it in this talk, but there is already the hybrid technology of PET and CT has been in existence for a long time, and it's kind of a competitor to the technology I discussed today. And so there you get radiation dose from both. You've got the CT and the PET giving radiation dose. And so one of the advantages of PET MRI is you only get radiation dose from one of the two components. And PET CT is essentially every PET scan that is done today is done with an accompanying CT. So one thing here is we could perhaps get rid of the CT in many of the cases and reduce the dose to the patients overall.
I have a remote question here, relates to the effect of the magnetic field on the avalanche photodiode. It says, what is the reduction in the APD signal due to the resistance increase due to the Hall effect?
Okay, so we do see the Hall effect, but in a different context. And I'm just going to scoot back to an earlier slide, because I didn't point it out when I showed it, and it is there. So it doesn't really affect the amplitude of the signal. What it affects is it affects this map of the detector crystals. So if you look, where's my pointer? Come here, pointer. Okay, the top left image is taken. Oh, there we go. The top left image is taken outside of the magnet, and so we see a map of our crystals in our detector. And you notice when we go inside the magnet here, it looks to rotate a little bit, and that's the Hall effect. Because of the orientation of the avalanche photodiode, what it's doing is it's giving a slight rotation to where those crystals appear in that image, and that is purely a Hall effect, just the magnetic force on the electrons. But we don't see a change in the gain, and it may partly be because of the orientation of the avalanche photodiode with respect to the magnetic field that we don't see a gain change.
Other questions? I think one of the, we have another one here. I've got one too, but let's go with this one. Great talk. Could you compare the signal-to-noise ratio of the avalanche photodiodes?
I'm not sure. Yes, I'm not sure what I'm supposed to compare them with.
Maybe to the photomultiplier.
Is there a... yes, versus the photomultipliers, and that's Steve Connolly, who I know well. Hi, Steve. So the signal noise is definitely worse. The gain is lower on the avalanche photodiodes. On the ones that we use, we get a gain on the order of about 10 to the 3 versus it's about 10 to the 6 for a photomultiplier tube. We cool our avalanche photodiodes to help the noise, and that helps quite a bit. So we cool down to typically around 0 degrees centigrade. But the signal noise is still inferior. So the exciting thing potentially now is the advent of a new technology called silicon photomultipliers that kind of combine the best of both worlds. So they have similar gain to photomultiplier tubes, but they're still silicon-based detectors that are immune to magnetic fields. And so many of us in the field are very excited about the possibility of those photodetectors for projects like the one I've just shown.
I think one of the systems you showed had about 16 detectors along the ring.
Yep.
How does the number of detectors or elements affect the resolution of the system?
Okay, so the smaller the elements are and the more of them that you have, the better spatial resolution you can get. Because you've got finer spatial sampling, and also you can identify where the photon interacted into a smaller volume. Eventually you run into some limitations because you start to hit other things being limiting rather than the crystal geometry, but we're not there yet. In fact, in our lab now, those crystals are about 1.5 millimeters in size. In our lab now, we've got down to some crystals that are about 220 microns. And there we're finally getting to the point where the physical size of the crystal is less the issue, and there are other physics, positron range, things going on that dominate the resolution you measure. So we could get better resolution we've shown so far by going to smaller crystals, and that's what we intend to do in the next generation. And then if we can put more rings and extend axially, we can get better sensitivity. We can capture more of the radiation, and we'll get more counts, better signal to noise. And so that's the other aspect of what we'd like to do.
And there's just one detector in each one of those elements, or is there a matrix of...
So each module, yes, well, there's one avalanche photodiode, but it's a position-sensitive avalanche photodiode, and there's a matrix of scintillator crystals on top of that. And so this is showing that matrix of crystals, so we can see each crystal corresponding to a spot, but it's one avalanche photodiode that reads that out. And the way it works is essentially the avalanche photodiode has a resistive layer on the back side, so that when electrons hit it, you get a charge division to the 4 corners of the avalanche photodiode. And by looking at the ratio of those signals, you can determine where the electrons were generated inside the device, and that's what gives you the position.
Mm-hmm. Other questions? We still have time for a few more questions. One over here again.
And this goes back to the knowledge of the spatial resolution changing across the field of view. Is there any plans to apply some type of DOI correction to the next generation?
Yes. So the question relates to the fact that for these ring geometry PET scanners, an effect you typically see is that the resolution is really nice right at the center of that ring. But as you move away from the center, the spatial resolution gets worse. And the reason it gets worse is that we have pretty thick detectors. We need that to stop these energetic photons, and we don't know how deep into the detector the photon interacts, and we end up with what's effectively a parallax error. And so at the center we're fine because all the crystals are pointing towards the center. But as soon as we move off center and photons start to penetrate obliquely into these crystals, we run into some problems. And so what the question was referring to is, do we have plans to develop a detector that has what's called depth encoding, so we can actually measure the depth of the interaction inside the crystal and get around this problem? The answer to that is yes. The next generation we are trying to do that, and the way we do it is we actually, rather than just putting an avalanche photodiode on the back end of the crystal array, we also put one on the front end of the crystal array. And by taking the ratio of the two signals, we can determine how deep into the array the photon interacted, and that's the solution to that problem.
Question?
From the perspective of the imaging software, you talked about how certain tasks are easier, how registration is easier, PET reconstruction is easier. Are there any new and unique challenges that counterbalance this, or is everything just easier?
Well, this system makes everything much easier because things are registered. There's certainly been tremendous advances on the software side over the last few years trying to address the problem if you don't have scans from the same imaging system and you need to register them, what's the best way to do that? So we have all these incredibly sophisticated warping methods that are constrained with some knowledge of the anatomy, and they get more and more sophisticated as we build in models for how different parts of the body can warp and move, and they continue to get better and better. But there's still just fundamental limitation that there's only so much information content in the images, and particularly in the PET images. The MRI is fantastic detail, but in the PET images there's only a certain amount of detail in there to grab hold of, to use with these algorithms, and that's what limits it rather than the algorithms themselves, which have become very, very good.
Anyone else? I'll ask the last question then. Back to the clinical applications. So you have the ability to look at the difference in a tumor of a metabolically active region and one that's not active. What again is going on there? Is that a high growth region compared to a low growth region? And also, what are the implications clinically for that? What does that information, that ability to look at the different parts of the tumor in this second way help you to do?
Yeah, so tumors are often very heterogeneous, particularly as they grow larger. So you tend to have regions of necrosis, you tend to have regions that are very viable and growing very aggressively. And so I think the advantage from a clinical standpoint as you start to treat these patients is being able to monitor what's happening and to really understand, and not just take whole tumor measurements that can be very, very misleading, but to really understand regionally what's happening to metabolism, to blood flow, to actually look at, if you can, to radiolabel the drug and look at, is the drug actually going to the viable part of the tumor? I mean, that's an important question. That's perhaps more of a research question, but still we know so little about the interactions between our treatments and then the tumor and its ability to then withstand those treatments and recur. And I think having these tools that can really look at this and look at both the structure and the function will help elucidate some of these things.
Thank you very much. Let's thank our speaker again.
Thank you.